Okay, hello everyone. I believe I'm now live. I see we only have 16 participants in the room as yet. So I wonder whether it's not useful to just hang in 1 minute so that people can connect, seeing that the session only started now. So let's just wait for 1 minute and then we get going. And I've seen you, Anina, so great, you're in the session. So Anina's our first speaker, but let's just hang on a bit. Okay. Doesn't seem like more people joining, which is surprising. Usually in a face-to-face conference in the pollination session. Yeah, so hello everyone. From my side as the chair for this session, I'm Sheriff Geerts from CPUT. And from two excellent plenary presentations, we now move into the different parallel sessions. And of course, this is the after lunch session, which tend to be the tough one. But we look forward to a number of really interesting pollination presentations, which will keep you awake. So just in terms of housekeeping, there's a Q&A box where you can put your questions. You can also vote for questions. So if you like a particular question, you can upvote it. So we can take those questions with the most votes. Unlike an in-person or face-to-face conference, I cannot indicate when your time is nearly up. So, and I don't want to interrupt a few times. So it's up to you to stay within your time or keep an eye on the clock. What I will do is when there's about 3 minutes left, I will interrupt and warn you as such. So it's 20-minute slots. So if you're still busy at 17 minutes, I'll just indicate as such. And then you can decide whether you want to wrap up quickly, have time for questions, or continue. Okay, so we have 4 presentations, and then we have a little break, and then we have another 3 presentations. The idea is to stay in sync to some extent with the other session. So we'll have questions after each presentation, and at the allocated time, so after 20 minutes, we'll try and move on to the next presentation. I do see that other sessions got one more presentation than this session. So there's always time to address those unanswered questions at the end of the session. And I think I've covered everything. Now let's get over to the presentations, and we start with the presentation by Anina on ericas and bird pollination. Over to you, Anina.
Thank you very much. Let me just get the screen share going. There we go. Right, so let's get my pointer ready. So I'm Anina Kutse from Nelson Mandela University, and I'll be talking about some of my work on reproductive barriers in erica that I did while I was at UCT. Now, this phylogeny here shows a species radiation where many species evolved over relatively short time periods compared to the more common process where species evolve over very long time periods. And you can see that it produces, this radiation specifically produced a lot of species over a very short time. And this phylogeny is in fact of the genus Erica. And the very large yellow clade that you see over here is the Cape Erica clade. And this radiation has produced nearly 700 species that now exist in the Cape Floristic region. And this radiation occurred in a small biome, the Cape Floristic region, and therefore there's a large number of these congeneric species that still need to coexist together, often sympatrically. And consequently, when these ericas occur together and when they flower together, there needs to be reproductive barriers that maintain the species boundaries and prevent genetic contamination and genetic swamping. So ecologists often distinguish between pre-mating and post-mating reproductive barriers. And in plants, these processes are most easily distinguished as pre-pollination and post-pollination barriers. Some of the most common pre-pollination barriers include segregation of species in space, time, or the use of different pollinators. And post-pollination barriers include a number of mechanisms that can occur after pollen has been deposited, and somewhere along this pathway, the production of the zygote is prevented, or the development of the offspring or their reproduction is prevented. Now, typically, multiple mechanisms are at play to effect reproductive barriers, and it's very rare that any of these mechanisms are 100% solid, so they're usually leaky. But in general, for plants, the pre-pollination barriers are thought to be twice as strong as the post-pollination barriers. Now, in ericas, there are many species use different pollinators to transport their pollen. And some of them use birds, some use insects, some use wind, and then a few of them use rodents. And this different use of pollinators is a very strong pre-pollination barrier. However, if we zoom in on one group, the bird-pollinated species, then a different picture emerges. There are about 67 species of ericas that have been formally classified as bird-pollinated based on the shape of the flowers as well as their nectar properties. But all of these species are predominantly pollinated by the orange-breasted sunbird, which you can see in this photo here. At least in the southwestern Cape, this pollinator is dominant. And so when they share this pollinator species, it creates the potential for hybridization between species. And so there must be some barriers that prevent or that maintain the species boundaries. So in order to investigate this, we looked at 4 populations of 3 different erica species, and we quantified the reproductive barriers. And here you can see our focal species, and each of them occurred with another bird-pollinated erica species flowering at the same location at the same time. And essentially, for each of our focal species, we calculated to what extent barriers can theoretically reduce gene flow between these species pairs. So firstly, we will look at the pre-pollination barriers. And the first one we looked at was geographical isolation. And large-scale geographical isolation was quantified with distribution data at 1/4 degree square resolution. And we calculated the percentage of area not shared with the co-flowering species. And that's produced these percentages here. And as you can see, it was quite low percentages for 3 of the species, which means that there's weak geographical isolation between these species pairs. For the one Erica coccinea species, there was a high percentage, but that was mainly because it occurred with Erica ebetina atrorosea, which has a very small range that's completely enclosed within the Erica coccinea's distribution. So although it's seems strongly isolated, it's mainly just because its co-occurring species has a small range. Secondly, we looked at phenology, and at 2 of the study sites, we quantified flower abundances for 6 months of the year, and then used this information to calculate phenological isolation. And what we found that for this species pair here, there was quite strong isolation because they shared a short period of flowering time. And for the one Erica coccinea population, the percentage was small, so there was weak isolation. And then for Erica curviflora, we did not quantify this formally, but the 2 occurring species peak their flowering at the same time. So that's probably also weak isolation between them. And then thirdly, morphological isolation is another mechanism that can be used. Now, in Ericas, the bird-pollinated species all have similar flower morphologies, these long tubular flowers, and the birds probe them in much the same way. And therefore, the length of the flowers could be an accurate indication of where pollen would be deposited on the sunbird's bill or feathers as they're probing the flowers. And therefore, that information about the size of the flower parts can be very valuable to determine whether pollen will be mixed between species as a sunbird visits 1 or 2 or more different species consecutively. So we we measured flowers, and we measured the female parts, the pistils, and the anthers, the stamens, the male parts. And then we calculated this distance between the male and female parts. Firstly, for flowers from the same species, and because you would expect that distance to be small so that pollen can be effectively transported to flowers from the same species. But you would expect that this distance between male and female parts from different species should be much larger to prevent the pollen being mixed between species. So we compared these distances between conspecific flowers and heterospecific flowers. And here are the results for the 4 study populations with stigma anther distances on the y-axis here. And what you can see over here is the conspecific differences on the left, and the heterospecific distances on the right, which are much larger, which means there is strong isolation between these species pairs. So that was true for these 2 populations. They experience strong morphological isolation. However, in these populations, those distances are quite small and non-significant, and therefore these 2 populations actually experienced weak morphological isolation because of the high risk of pollen being mixed by pollinators. So to summarize the pre-pollination barriers, here are the 4 study species again and the 3 barriers that we quantified. And as you can see, many of the pre-pollination barriers are in fact weak, and that suggests that strong post-pollination barriers are required in this system. So we also quantified post-pollination barriers. And we did this by doing hand pollination experiments in natural Erica populations. And basically, we wanted to determine whether the heterospecific pollen can produce seed or whether it reduces seed set significantly. And here are the results for the 4 populations. I'll just focus on this one first. On the right hand here, we have the natural treatment, which was basically flowers left unmanipulated and left to be pollinated naturally. Whereas here on the left is the outcross treatment, where we added lots of pollen. So it's basically simulating optimal pollination conditions. And we compared these 2 to this heterospecific pollen treatment, where we added the pollen from Erica plucaneti to Erica coccinea flowers and then quantified the seed set. And as you can see, the seed set is significantly lower compared to these other two treatments, which supports the hypothesis that there is strong post-pollination barriers in these species. And the same was true for all four populations that we studied. Nonetheless, the application of pure heterospecific pollen significantly reduced the seed set. So that means there's a very high fitness cost, and therefore there's still incentive or selection for the plants to evolve pre-pollination barriers as well. But what if flowers still receive conspecific pollen after receiving heterospecific pollen? So we simulated this in one of the populations where we added heterospecific pollen to the flowers, and then 3 hours later added conspecific pollen. And the result can be seen here, where the seed set for this treatment was still lower than that of outcross treatment. And that means that there's still a very high fitness cost, even though that can produce more seed compared to a pure heterospecific pollen load. And this also suggests that there's this form of reproductive interference would also drive selection for pre-pollination barriers that will prevent that heterospecific pollen transfer. And now, if there is selection for pre-pollination barriers, then this selection will be strengthened by pollinator dependence of these plant species, because the plants would then respond more strongly to selection by pollinators. And so to determine whether there is this dependence, we conducted self-pollination and self-autonomous pollination experiments. And the results are just added here to the graphs I showed earlier, with the selfing treatment over here and autonomous selfing over here. And in 3 of these populations, we found that these species were self-sterile, which means that they are probably very dependent on pollinators for their reproduction, and therefore they are likely to respond strongly to selection by pollinators. So to wrap that up, in this Erica system, we found weak pre-pollination barriers. However, we found strong post-pollination barriers. And so this can effectively facilitate the coexistence of the species. However, although this barrier is strong, there were still some factors that reduce the fitness because it only acts late after pollination. And so these factors that reduce fitness could still drive selection for pre-pollination barriers, such as, for example, traits like flower color or nectar or flower morphology that could help in pre-pollination barriers. So in conclusion, these ericas had weak pre-pollination barriers, which is actually relatively rare in plants. And it makes it a fascinating puzzle to understand how this large amount of ericas can coexist. And our evidence suggests that it's because of strong post-pollination barriers that they are able to do this. But there are, of course, a lot of barriers that we haven't explored yet, such as, for example, pollinator preference and hybrid sterility and things like that. So hopefully this can be investigated in the future. And then I'll just finish off by thanking everybody and the organizations that made this possible. And thank you so much for listening.
Yeah, presentation. So let's go over to the questions. I believe you can also speak up. I think that can work. So if you prefer to rather ask your question okay, so let's have a look at the question box. Glennis asks, Did you measure the reproductive part at a specific stage of development? And if so, what stage was it?
Sure. So we measured mature flowers. We don't know exactly how long these Erica flowers last, but we can tell whether they've been visited or not. So we did measure flowers that were mature, which you can see from the how the anthers have developed, and before they start showing signs of senescence.
Okay. Thanks, Anina. Just a question from my side. So looking at all these barriers, and especially the pre-pollination, but also post-pollination barriers, these are not perfect. So I would expect to see more hybrids in the field. I've never looked for them really, but are there hybrids in the field more than you would expect, I suppose? I don't know how to phrase it, but I would expect, based on what you've shown, that there are more hybrids than I've seen. What are your thoughts on that?
Yes. So there are some hybrids around. I've seen some of that in the literature. I'm not an expert, and Ericas are so difficult. So, but there are, but I think it is actually not common. And yeah, so it is quite still quite puzzling to understand why we don't see those hybrids and what exactly are the barriers. And I think it differs among the species probably as well, which combination of barriers are operating in that system. But in general, there are actually more hybrids, and hybridization is possible more frequently than we actually think. We tend to think that barriers are very solid, but they're not that solid.
Okay, so in effect, you could add up the different barriers. So you indicate them as weak and strong, but I suppose it's more continuum. So you can add them up for each species and see which ones are most likely to produce hybrids in the field.
And of course, the seeds, we found seeds, but we don't know how fit those seeds are. So they could be at the later stages of the plant development. There could be a lot of things that prevent it from being fit.
Yeah. I suppose the next step is to germinate those seeds.
Yeah.
Look at that. Thanks a lot, Anina, for that presentation. We need to move on. So from birds to the bees, the next presentation by Cobalt, I hope I pronounced it correctly, is on pollination in macadamias. Let's just see if
I hope you can all hear me.
Yes, we can.
Okay, fantastic. I'm just getting my presentation ready. Okay, I hope you can all see. Hi, can anyone hear me? I can't hear anybody else. I don't know if this is a problem or not.
You're good to go. Joe,
Okay,
you can
fantastic.
continue with your presentation.
Okay, great. Thank you. Okay, so my name is Joe Cobbold, and I will be addressing Shakespeare's most unanswered question today, to be or not to be. The full title of my project is To Be or Not to Be, an analysis of floral rewards and pollen transfer efficiency in South African macadamia. So to be or not to be is South Africa's 4.8 billion rand problem. Let me explain why. South Africa, macadamia is grown all over the world, but South Africa has been the top producer of macadamia for the past 4 seasons. If we look at the diagram on the bottom left, we see that each macadamia tree can produce between 2 to 3 million flowers per tree. Only 75,000 of these flowers actually end up producing a nut that is harvested at the end of the day. We therefore see that pollination is only successful around 0.3% of the time. When we compare macadamia yields to the yields of maize achieved by South Africa in the 2020 season, we kind of get an idea as to how valuable macadamia nuts are. So South Africa produced around 49,000 tons, worth around 4.8 billion rand, and we produced just under 15 million tons of maize, worth about 50 billion rand. As I said, this just kind of gives a person an idea as to how valuable macadamia is for our industry. So the interaction between bees, pollination, flowers, and pollinator rewards is actually a very simple cycle, but it's a lot more complicated than that, actually. I will be specifically focusing on those sections highlighted in red, as those are the parameters that I measured during my study. When looking at bees specifically, they were chosen as the pollinator of choice because bees are often brought into macadamia orchards during the flowering season in an attempt to improve macadamia pollination, leading to a higher yield. So, what do bees want? Bees often, well, they collect pollen as well as nectar, specifically with a concentration of between 50 to 65% sugar, and they would obviously prefer to be able to forage easily. When looking at pollination, pollen transfer efficiency was measured, and this is defined as how much pollen moves from one flower to the stigmatic surface of another flower successfully. This was measured to give light to quite a prevalent argument between macadamia farmers. Some farmers believe that it is financially justifiable to bring in kept hives into their orchards during the flowering season, whereas other farmers feel it's okay to only rely on natural pollinators. The sepal surface of the macadamia flower was also analyzed. There's obviously an interaction between bees specifically and the actual sepal surfaces of a macadamia flower. And I mean, it would kind of be logical that the easier it is for a pollinator to interact with the flower, the easier it would be for the pollinator to pollinate the flower. Pollinator rewards were also analyzed, pollen and nectar specifically for different cultivars. And then my project had two different aims, each with their respective objectives. My first aim was to determine macadamia pollinator rewards and floral accessibility. My objectives were to measure pollen production, nectar volume, nectar sugar concentration, as well as sepal surfaces of the different cultivars. And then my second aim was to determine if having kept hives improves pollination versus only relying on wild pollinators. This was done by measuring pollen transfer efficiency. For my materials and methods, I had two different collection sites. I worked in Mpumalanga and KZN respectively. In each of the provinces, I had two farms. One of the farms had kept hives versus a different farm in the same area that did not have kept hives. I also did collections at the Agricultural Research Council in Nelspruit. 15 different cultivars are analyzed during this project, 8 of which are commonly planted in South Africa. And then to quantify the amount of pollen produced by each cultivar, collections were done in the field into glutaraldehyde. The flowers were dissected and the pollen stained with basic fuchsin. Slides were prepared and then analyzed under the microscope. 6 individual photos were taken per microscope slide and then stitched together using imaging software. These stitched images were then run through ImageJ and to count the pollen grains. The parameters for ImageJ were also verified with hand counts. To measure nectar volume and concentration, specifically, I used microcapillary tubes. The flowers were first drained and then bagged, and then 12 hours later, I measured the amount of volume, the volume produced by each flower. For nectar sugar concentration, I used a handheld refractometer to do those measurements. When doing the pollen transfer efficiency sampling, I did these specifically in the 695 and 816 cultivars. On the farms that had kept hives brought in every year, the sampling began at a tree closest to where the hives were placed. And then a single flower was collected every second tree moving away from the hives. Exactly the same format of sampling was done on farms that did not have the kept hives, except sampling began at an area of natural vegetation that could harbor potential natural pollinators. Pollen transfer efficiency, the pollen was then counted. As I mentioned, the flowers were collected into individual Eppendorf tubes containing 70% ethanol. The slides were then prepared as the same method as explained before and photographed under the microscope. Again, the images were stitched together and run through image J. 2 different pollen counts were done for each individual flower. As I said, the pollen in the loose pollen within the Eppendorf tubes was counted, as well as the pollen that occurred on the stigmatic surface of that flower. Those 2 values were then used to calculate a population PTE percentage for each of the farms. In order to analyze the sepal surfaces of each of flowers of each of the different cultivars, scanning electron microscopy was done. Field collections were done into glutaraldehyde. The flowers were dissected and taken through a dehydration series. They were then critically point dried and mounted onto SEM stubs. Photos were then taken using the SEM, and a visual analysis was done of the photographs taken. For my results, the mean pollen counts per cultivar I did in ANOVA. Here we can see that there is a significant difference between the cultivars, but not between the flowers themselves, which made up which were the biological replicates. As I mentioned previously, there is a significant difference between mean pollen counts per cultivar. You'll see that there are 3 different color sets of arrows, if I can say it that way. The blue arrows indicate 2 cultivars that scored within the highest mean pollen count group, those cultivars being 705 and NG18. The 3 green arrows designating cultivars 814, N2, and Own Venture were the lowest scoring mean pollen count group. If I can also draw your attention to the pink arrows being cultivar 695 and 816. 695 scored above average in terms of its mean pollen count and 816 below. Mean nectar volume was not a significant parameter. And here, if I can again draw your attention to 695, that scored way below average, and 816, that was slightly above. When looking at mean nectar sugar concentration, this, however, was a significant parameter. And I again have the arrow groupings with the blue arrows indicating a group where within this blue arrow group, there was no significant difference. But when compared to all of the other cultivars, there is. Again, the pink arrow 695 scored above average. But when a person looks at 816, we see that 816 scored far above average, and it is also one of 3 cultivars that scored a mean nectar sugar concentration that falls within the desired sugar concentration range preferred by bees. When I analyzed the images, the same images taken of the sepal surfaces, I chose cultivar 849 as a cultivar to represent 14 out of the 15 different cultivars. This was because these 14 cultivars, their sepal surfaces all look relatively similar, much resembling this image depicted above. Dado, however, was the outlier cultivar, as a person can see that there is quite a big difference between the 2. 14 out of 15 different cultivars generally had flattened sepal surfaces, and they were slightly striated, whereas Dado had papillae and it had dense striations, lending towards a rougher sepal surface as opposed to the other 14 cultivars. When we looked at the mean PTE of the different pollinator populations, we see that the PTE value for farms that brought in kept hives was generally quite a bit higher than the PTE for the farms that did not bring in hives. When we split those PTE values into the 2 different cultivars that I collected from, we also see that 816 contributed a lot more towards the PTE value that I showed previously, as opposed to 695. When measuring pollination success and PTE value, we do also see that there's quite a strong correlation between the 2. For a discussion of all of the floral rewards together, I basically developed a color system. So the darker the blue, the higher that parameter scored during my entire study. I generally also only focused on cultivars that are already commonly planted in South Africa, designated in the top blue in the first row, as you can see. So what we are basically looking for is a column that is dark blue, well, the darkest blue possible all the way through. So when one does this analysis, we see that 695, 788, and A4 are three cultivars that generally score quite high in all of the parameters measured. This was also kind of backed up by a study that Macadamias South Africa did of all the different cultivars planted in the 2020 season. So 33% of the trees planted in the 2020 season were from the 695 cultivar, 3% were from the 788 cultivar, and 29% were from the A4 cultivar. When we look at the PTE, we see that overall, the PTE on farms that bring in kept hives excuse me, was much larger than the PTE on farms that did not bring in kept hives. This can simply be explained by the fact that on farms where there are more bees, there are more pollinators and more pollination is occurring, therefore leading to a higher PTE value. This was also kind of confirmed and backed up by a statement released by Dr. Hanley Human from the University of Pretoria. She stated in a publication that pollination does depend on pollinator populations, and any decrease in pollinator populations will lead to a decrease in yield. When a person looks at the different cultivars that are analyzed for the PTE specifically, we see that 695 scored a much lower PTE as opposed to 816. This can simply be explained as well. When I did the analysis, we noticed that 816 scored nectar sugar concentration within the range desired and preferred by bees. This could lead to a phenomenon known as floral constancy. That generally occurs in bees. So when bees find a good food source, they generally tend to stick to it. So if 816 had a good nectar sugar concentration that they preferred, they would generally have stuck to that cultivar. Cultivar flowering time also makes quite a big difference. When I was able to do my collections, 816 was flowering optimally, and 695 would only have come into its peak about 2 weeks after I left. And again, when correlating PTE and pollination success, we see that there is a strong positive correlation, and the farms with hives all scored above the regression line. When discussing 816 and 695 specifically, we see that both cultivars are commonly planted in South Africa. 695 scored much higher in terms of the amount of pollen produced by each of the flowers, whereas 816 had a higher nectar volume, although that was not a significant parameter. 816, however, did also have a more ideal nectar sugar concentration as opposed to 695. The sepal surfaces were relatively the same, and 816 scored much higher in terms of its PTE value. So in conclusion, an analysis of floral rewards allowed for informed cultivar recommendations based on pollinated needs. So at the end of the day, to be or not to be, I would personally rather be than not be. I feel that bees would improve pollination to the point where it is financially feasible for farmers to bring them, to bring hives in onto their farms during the flowering season. As my list of references, and I please need to also acknowledge these people, many of which this project would not have been possible without, especially those designated in bold. And again, a big thank you to Macadamias South Africa for funding my project. Are there any questions?
Much for a lovely presentation, and apologies, my signal just cut out over the results section, so few slides. Thank you very much. And this seems like a really good study species, especially if you look at something like seed set. It's at least then something you can eat. So I don't know if you touched on that.
Indeed.
Okay, so let's go into the Q&A. I see there's one question coming up. Is a rough sepal surface easier to handle by bees?
I would say so, yes. I mean, the easier it is for a bee to physically hold onto the flower, the better it can interact and pollinate the flower. So yes, and studies have shown in other species that the easier pollinators interact with the flowers, the higher the pollination is at the end of the day. So yes, I would say the rougher the surface, the more pollination you could potentially get at the end of the day. Obviously, more research would need to be done, but yes.
As always, yeah.
As always.
People, please feel free to post questions in the Q&A. It's nice if you don't, because then at least I can ask a question. But please go ahead. So just a question in terms of other pollinators. Apologies if you did mention it, but I looked at the thesis once. I think it was a student from Stefan Voort. And I think honeybees only made up about 60% of visitors to macadamia flowers. So how important are other pollinators other than honeybees?
I didn't really look at other pollinators specifically. When I was developing the project, I approached individual farmers and I said, look, what are the problems that you have? And they largely only mentioned honeybees because it's a pollinator that can currently be controlled by bringing the by bringing the hives in and out. So I do know that, as you obviously said, that there are many other pollinators that are incredibly important in the macadamia industry, but I did not focus on them specifically. If I'm not mistaken, I'm pretty sure. Oh no! I think I might be thinking about something else now, actually. But yes, sorry, I don't have a faster answer to your question, unfortunately, but yeah, because I focused on the honeybees.
You can't look at everything in one project, eh?
Unfortunately.
But something to just think about, because honeybees are only 60% of the pollinator visits. Yes, what is the other 40%, and where do they come from, and how important are they? Especially for some of those with lower honeybee visits, maybe they receive more visits from other groups. Okay, let's go back to the Q&A. Glennis asked, did you look at pollen viability at all?
I unfortunately didn't know. So I've got no idea in terms of that, to be very honest with you. But again, perhaps a point of future research.
Yeah, an important one. Yeah. Okay, let's do one more. I think there's only one more. Anyways, could farmers select cultivars with the best rewards? Or do they have poor quality and other traits, such as nut quality?
My project was centered around obviously pollination biology, so I unfortunately didn't go as far as nut quality, and because of the macadamia flowering season, it's not conducive to finish within one honors year, so I unfortunately couldn't look at nut quality as well. But from what my research showed, a person could select cultivars, again, only according to honeybee preferences. A person does obviously have to consider other pollinators as well. But yes, from what I've seen, you can make recommendations based on honeybee desired rewards. So yeah.
Okay, but really well done, and it's nice to see such applied pollination projects. We need those. So and really well done for an honest project. It's
thank you.
Okay, thanks.
Thank you.
So then we go from macadamias. We move on to seed dispersal by monkeys in the Amaryllidaceae, and over to Butler. Sorry, I didn't get all the names before, and apologies for that. But there we go. Anna, then it's over to you. Thank you.
Hi.
I can hear you. You can just share your screen.
Right.
Okay, great. We can see the screen. You can go ahead.
Perfect. Okay. Hello, everyone. My name is Hannah Butler, and today I am going to be talking about seed dispersal by monkey spitting in Scleroxis, fruit selection, dispersal distances, and effects on seed germination. So just to wake up all of you pollination biologists that are sitting in this session. I'm going to be talking about seed dispersal. And as much as pollination is important, seed dispersal, I would argue, is also an important part of a plant's life cycle, as it is the culmination of reproduction. Seed dispersal helps to maintain populations as well as to establish new ones, and is essentially the only way that plants can move throughout their life. The evolution of the development of fruit, although potentially for different reasons initially, has resulted in multiple dispersers being used, and this has led to the diversification of angiosperms as a whole. If we look at fruit characteristics, this can tell us more about the seed disperser. So for example, if we have a look at this amaryllis fruit that I've shown here, you can see that it's the fruit is split open. There's no bright color, there's no fleshy fruit, and the seeds are really thin and lightweight. So we can assume that they are dispersed via wind. Fruit processing by a seed disperser can affect seed dispersal in multiple ways. An immediate factor would be distance. So this is essentially how far the seed has been moved. Post-dispersal factors affect seedling recruitment. So this is seed survivorship, seed germination, as well as seedling growth. I work in the family Amaryllidaceae, which, lucky me, it's my favorite family. And there has been some work on the subfamily looking at seed dispersal. Sneeman and Linder looked at the subfamily Amaryllidae and classified them into a nemogeochory, which means tumbling, such as your Brunsvigia, a telochory, which is rainwash, autochory, self, and a nemobolist, which is wind. And this means that all of these are wind and water dispersed. But Keviel and Johnson had a look at the genus Clivia and found that they were dispersed by monkeys. Now, Clivia is in the subfamily Hemanthae, and Hemanthae is characterized by having baccate fruits, which means it's a fleshy drupe, and that means that the endocarp is fleshy, and they're generally brightly colored, the fruits. So we would therefore assume that this is zoochory, right? Dispersal by animals. Again, the fruit characteristics tell us something about the seed disperser. However, there are a few puzzling parts of this. There are large, hard seeds. They lack dormancy. They cannot survive gut passage, and they are poisonous. I looked in the genus Scodopsis to answer some of these questions. This is an amaryllis genus of 9 species found throughout Eastern Africa, and it's pollinated by butterflies or by birds, as my previous research has shown. And they also produce these brightly colored fleshy fruits with large toxic seeds. And again, we have to ask the question, are they dispersed via animals? Zoochory. Well, I looked at 2 species, Scodopsis puniceus. This species is widely distributed and found in forest patches and bush clumps, and Scodopsis multiflorus subspecies Catherinae. This species is restricted in its habitat to swampy areas surrounding streams within coastal forests. So we set up some video traps on the infractescences, and this is what we found. So this is a vervet monkey interacting with the fruits of Scodopsis multiflorus subspecies Catherinae. Now we also saw some birds. However, it seemed that the fruits were kind of too large for them to handle properly. So they would either drop the fruit in entirety, or manage to grasp a little bit of the fruit off it, so probably don't result in seed dispersal. For Scodoxus puniceus, we also found vervet monkeys. So if you look closely, you can see that the seed dispersal mechanism is monkey spitting. So first of all, the monkey arrives, it picks off the individual fruits, pops it in its mouth, where it removes the fleshy endocarp, and then it spits out those hard, toxic seeds. We had a look at fruit selection, and you can even see this in the videos that I showed. Monkeys clearly selected the red over the yellow over green fruits, as shown in the figure. So we can say quite clearly that monkeys are attracted to the bright color. We had a look at seed dispersal distances, remember that's an immediate factor, and found that the majority of seeds were dispersed further than what we measured. If we have a look at the figure, so A and B is Scodoxus puniceus, and we measured up to 2 meters, whereas C is Scodoxus multiflorus subspecies Erythrina, where we measured up to 1 meter. And as I just said, those bars right at the end for further than we measured are the highest. But there is some short distance dispersal, and this may help to maintain populations. We suggest that those that were dispersed further was via cheek pouching. Now, cheek pouching happens when the animal, so the monkey, is disturbed, the troop moves on, but the monkey keeps those fruits or those seeds in its cheek pouches, and then it'll spit them out further away. Now, this is really useful for the monkeys as it helps to reduce vulnerability to prey and increases feeding efficiency because of less competition. We then looked at a post-seed dispersal factor, which is seed germination. And we found that color did not affect germination. However, the pulp removal strongly promoted germination. So if we have a look at the figure, the colors represent the fruit color. The circles are the ones that are depulped, so where the fruits have been removed from the seeds, whereas the triangles are intact. So you can see that within 2 weeks, 50% of the peeled seeds had already germinated, but it took about 5 weeks for 50% of the unpeeled seeds to germinate. So this pulp removal that the monkey stud does is really important for germination. Are there any other monkey spitting dispersed plants outside of the subfamily? Well, it turns out that they're a really important part of diet within African species as well as into Asia. There's also a combination of the short and long distance dispersal, but this is usually under the same tree crown as they're normally tree species. And our work on Cultivar as well as on Scodoxus is potentially the first time that we're seeing this system within bulb. They also found that they moved before spitting, which again gives us evidence for cheek pouching. Now, if you have a look carefully at this picture of a different monkey seed dispersed plant, you can see that the fruits are this bright color, they're fleshy, and they also have all these bulges, which are the large, fewer seeds. And this leads to better for cheek pouching, as they're much easier to keep in their cheek pouches and much harder to swallow. They also found that depulping also increased germination. In conclusions, monkeys disperse the seeds of 2 Scodoxus species via spitting. And both plant species occur in habitat niches that are frequented and populated by monkeys. This is known as directed seed dispersal. Because they are in the same population, they're in the same space, and then move to the same space. So the seeds are being dispersed to a specific favorable habitat, which means that seedlings have a higher probability of survival. So monkeys are really important for seed dispersal in the genus Scodoxus. They're attracted to the fruits. They then depulp the fruits, which leads to faster germination, and then spit out the seeds, which is the dispersal mechanism. Now, a quick note on my favorite photo that came out of the study. This is in fact a Simango monkey. As I said before, the majority of the monkeys that we saw were vervet monkeys. But when we looked at Scodoxus puniceus in a forest environment, we found that it was more likely that Simango monkeys were in the same area and would do the same job. But Simango monkeys are known to be quite elusive, and we only got a few images, and this absolutely perfect one of it staring straight at us. It is likely that related fleshy fruited species within the same subfamily, Hemanthae, also rely on zoo quarry. The pictures that I've shown here are from my work on Hemanthus deformis, so a related species in the same subfamily. And here we are finding that they are dispersed via rodents or birds, so probably different from monkeys because they occur in different habitats, as well as having those smaller fruits. And we're also finding very similar results, that they germinate faster after being depulped, and that there is a short as well as long distance dispersal. If you look carefully at the picture on the right of the rodent, you can see it carrying a fruit back to who knows where, beyond the camera anyway. Thank you to Steve Johnson, my supervisor, and the UKZM Pollination Lab, as well as everyone who's helped me along the way. And I hope I didn't rush through all of my time. Thank you.
Excellent. Thank you very much. And some really nice video footage there. It's always tricky to get good video footage from so close up. So well done. Really interesting. We have a few minutes for questions. So let's, please post them in the Q&A box. From Glennis, thanks, Hannah. An interesting talk. Does time to germinate really matter? If so, why? And I suppose this question comes from the fact that in the end, germination rate was similar between all treatments. The one was just germinating faster than the other one. So maybe you can shed some light on that.
Well, the seeds lack dormancy, so they have to germinate pretty fast, and just having that competitive advantage because they germinated faster could probably really help.
Okay. It's only a few weeks, so I suppose we need to have a closer look at the ecology of the plant and where it germinates and what are the requirements to answer in some more detail.
Well, of course, if you have a seed that is not surrounded by a fruit, it's more likely to be damaged from all sorts of things such as seed borers, etc. So it may be that a foster germination can help to escape that as well.
Just one. I don't see any questions. Just one question from my side. Just in terms of the colored fruits, if I remember correctly, monkeys take yellow fruits 50% of the time. So that's quite high. So you said they prefer the red ones, but it seems like they don't mind the yellow ones because it's 50% of the time they take the yellow ones.
Yeah.
What are your thoughts on that?
Well, at first we thought that it had to do with ripeness, and it probably does, but considering the germination, I guess it doesn't matter too much. So it's more about the attracting the animal to the plants. But interestingly, for the Cultivar research, they found that they didn't care about color at all and would go even for the green fruits. So, yeah, it's quite interesting that they didn't go for the green fruits at all, but they still did choose, the yellow.
Okay, let's see if there's more questions. Okay, there's some more. So if seed germination is not affected by fruit color, does it mean to say that seed maturity is not synchronized to fruit ripening? So seed maturity not synchronized.
I mean, apparently not, because from our germination project, we saw that it really didn't matter. And we've seen this for a couple of species now.
Yeah, and that makes the argument a lot stronger. But it's interesting how that actually then works. Let's do another question. Very interesting study. Do you perhaps know the enzymes in the saliva of the monkeys, especially in those that have been tick pouched, would affect germination rate?
This is an interesting question, and I've been asked before after I completed the study, because it makes me think I should have gone back and sucked the seeds before putting them in. So unfortunately, I have no idea. Possibly.
Okay, but
we didn't suck any seeds or take any from monkeys, so they still germinated quite well from just depulping the fruits.
Okay. Let's end up with this one. Any ideas if the fruit are edible for us? So the monkeys eat them. Is it edible for us as well?
I have been told that they were poisonous, but I was told that after I ate the fruits. So I'm still alive. So maybe take it in small doses, but yeah,
that's always advisable. Start with one.
Yeah.
Okay, great. Thank you very much. And a really interesting study system.
Thank you.
Thank you very much. Okay, so now for the last presentation before we take a quick break, we're back to pollination again and back to the birds again. And there's our speaker. Welcome, Lauren.
Thank you.
Over to you.
I'll just share my slides.
Yes, I'll just stay to confirm that.
Okay, is that all good? Can you see it?
Yes, I can see and I can hear you clearly. You can go ahead.
Great, thank you very much. Okay, good afternoon, everyone. My name is Lauren James, and I'll be presenting research which I did as part of my honors project at Rhodes University. And my title of my talk today is Contrasting Patterns in Flowering Phenology Between Species Adapted for Pollination by Generalist compared to Specialist Flower Visiting Birds. Plants that are pollinated by birds tend to have many of the same floral characteristics. These include a tubular or salviform or brush-like flower structure, bright colors such as red or orange, or other contrasting colors such as yellow or pink, a large volume of nectar, a lack of scent, and having a robust flower structure that allows birds to have a place to perch. And collectively, these traits form the bird pollination syndrome known as ornithophily. Now, there's evidence for there being a division within this syndrome into two separate classes of bird pollination. The first being specialist bird pollination, which is pollination by birds such as sunbirds and sugarbirds, which have got small bodies and long, thin bills. The flowers that they pollinate tend to have long, narrow tubes, and they have a smaller nectar quantity, which has a higher concentration of sucrose sugars. The second class of bird pollination is generalist bird pollination, and the birds that pollinate these flowers are larger and got shorter bills, and these are birds such as weavers and drongos, orioles, and bulbuls. And the flowers that they pollinate have a more open flower structure with exerted anthers and stamens. They tend to have a larger nectar quantity than specialist bird-pollinated species and a higher concentration of hexose sugars and richer, and the nectar is also richer in amino acids. So it's very important to consider multiple factors when studying the evolution of the mutualisms between birds and flowers that they pollinate. And so far, most studies that have compared generalist and specialist bird pollination have focused on a single floral taxon, and they have mostly looked at the morphology of the flowers in birds and at the nectar traits. But are there other factors that are important in these mutualisms apart from physical floral traits? And could flowering phenology, which is the timing of flowering, be an important floral trait that is adapted to pollinators? So there have not been many studies done in South Africa, as I've said, comparing these two classes of bird pollination. But from the few international studies that have been conducted, the main difference between these two groups has thought to be attributed to the different nectar requirements of generalist compared to specialist birds. Specialist birds feed predominantly on nectar, and it's a very important food source for them. The flowers that are pollinated by them are therefore thought to have their flowering time staggered throughout the year between different species. And this would prevent competition happening between species, prevent hybridization, and increase pollination efficiency. At the same time, it would be providing a source of nectar year-round for their pollinators. And this is a form of facilitation known as pollinator support, when collectively they are maintaining shared pollinators. The second group, pollinated by generalist birds, the behavior of generalist birds is quite different to that of specialist birds. They're much more opportunistic nectar feeders because they are omnivorous and therefore they only feed on nectar when other food sources are scarce. And therefore, to make most efficient use of these pollinators, it is thought that species pollinated by generalist birds would flower in late winter or the dry season when these pollinators have got a shortage of other food sources. And so the mass flowering of these species in winter would collectively attract their shared pollinators, and this is a form of facilitation known as pollinator sharing. So my hypothesis was that flowering phenology is a floral trait adapted to specific pollinators, with specialist bird-pollinated plants having year-round temporal partitioning of flowering, and species pollinated by generalist birds flowering simultaneously in late winter. My study was conducted in the southeastern summer rainfall region of South Africa. To start off with, I compiled a list of bird-pollinated species, and I did this by going through literature to see records of species that have been classified as bird-pollinated, and then also using the traits of bird pollination uniformly to determine whether or not a species was likely to be bird-pollinated. And I came up with 143 different species in my study region that were potentially bird-pollinated. I then divided this list into specialist bird-pollinated species and generalist bird-pollinated species. And this was again based on literature predominantly, and then also on observations, so photos that showed birds visiting flowers and having contact with reproductive structures, and therefore morphologically kind of fitting to be able to be an effective pollinator. Where neither of these sources of data were available, I used the floral traits that are charismatic of these particular classes of pollination. And so of the species I had enough data to analyze, I found 69 specialist bird-pollinated species, which were from 17 families, and 20 generalist bird-pollinated species, which were from 4 families. And this made a total of 69 species, which was 62% of the 143 bird-pollinated species in total, which I found in the study region. My next step was to get flowering phenology data. So this is the date of flowering, and all my records were from sources that had flowering scored as present. And my main sources of data were from the Botanical Database of Southern Africa through SAAB, and from citizen science records on iNaturalist. I also got additional data from TreeMap through the Virtual Museums Project, the Salmarsh-Shonland Herbarium, and also a few field observations to boost records. Now, flowering phenology is a cyclical phenomenon that goes through annual cycles, and it therefore does not have a distinct 0 point or value, or a start or a finish point that we can clearly define. And this makes it so that linear statistics are not appropriate for phenological trends. And it is therefore necessary to use a branch of statistics called circular statistics. So what I'm showing here is the flowering times of Erythrina humiana. All these blue dots are individual records of an individual flowering of the species, and that is going from January at 0 to December. And we can see here that the linear statistical mean falls very much not in the peak flowering time, which would rather be in the early part of the year in January. And using circular statistics, we can see that our mean is a much more accurate representation of the peak flowering time of the species. So to conduct my analyses, I used the circular package in R. And how it works is that you convert your dates of flowering into degrees of a circle that are then plotted around the circle. And the distribution of the data can then be tested using the Rayleigh test of uniformity that uses the resultant vector to give an indication of the concentration or the spread of the data points around the circle. And so the length of the resultant will give an indication of how spread out or peaked the flowering of that species or that class is. The mean angle, which is shown here in orange, gives an indication of the time of year of flowering. And so then to compare two distributions of two different plots, I use the Watson's test of homogeneity, which tests whether two distributions are likely to be drawn from a common distribution or not. So these are my results. This is for specialist bird-pollinated species, and on the right is for generalist bird-pollinated species. Each point plotted around the circle is the mean flowering time of a species, and the number of species is also indicated by these segments. And what we can see here is that the specialist bird-pollinated species had different species really scattered around the year in their mean flowering time, whereas generalist species tended mostly to flower in winter and spring. There is the exception of these 4 species here, but I will come back to those later. I then plotted all the flowering records. So rather than just finding the means, I plotted each individual flowering record of all species. And this gives us an interesting ecological interpretation into the results because it shows the overlaps between species in their flowering rather than just looking at the mean flowering time. And these results clearly show the patterns and how they differ between specialist and generalist bird-pollinated species, with the specialist bird-pollinated species having flowering clearly all year round, and generalist bird-pollinated species having a much higher instance of flowering in winter and spring. And though these two plots both do show that there is a significant peak in flowering in late winter in generalist bird-pollinated species and in late spring in specialist bird-pollinated species, we can see that the peak in the specialist is very weak because it has a very low R value. R value of 0 indicates that there's a uniform distribution of data around the circle, whereas an R value of 1 indicates that all the data points are at one point. And so we can see that the distributions are quite clearly different, and this is confirmed by the Watson's two-sample test of homogeneity, which shows that there is a clear difference in the distributions between these two groups of bird pollination. So significant differences were found in the patterns of flowering phenology between generalist and specialist bird-pollinated species, and this provides further support for generalist bird pollination and specialist bird pollination being recognized as 2 distinct classes of ornithophily. So flowering phenology can be therefore defined as a characteristic of a pollination syndrome. And this is important for understanding the evolution of these relationships, because if we think about it, in winter, when flower resources are scarce, that is the time when generalist birds would be the most effective pollinators, and it therefore makes sense for selection for generalist bird pollination in winter. Whereas in summer, when they're eating many other food sources, they're very unreliable pollinators, and therefore selection for generalist bird pollination would not be as strong. There are, however, other phenomena that influence flowering phenology, such as seed predation avoidance, the timing of seed maturation, herbivory, phylogenetic constraints and the climate. So looking at phylogenetic constraints, if we look at these Catanthus species, they do all flower in spring and early summer. And the two Leonota species that I investigated only flower in autumn. However, when we look at a genus such as Aloe, we can see that they follow the same general trends as the species overall, with species distributed throughout the year in specialist bird-pollinated species, and species only flowering in winter and early spring in generalist bird-pollinated species. A similar trend to the aloes can be seen in the genus Erythrina, with again specialists flowering at a scattered time throughout the year, and the generalists flowering only in winter. And then I just wanted to point out this interesting morphological change throughout the year in Erythrina, with going from January with Erythrina humiana here on the left, across to Erythrina zyri, and this is the order of flowering throughout the year. And you can see how the shape of the flowers changes, with these two open ones in the middle being the generalist pollinated species. Climate is another important factor that must be considered with flowering phenology, because it can definitely impact the seasonality of the environment, such as at a place with high altitude or high latitude, or in places with very strict dry or wet seasons. Flowering phenology will be constrained due to resource availability, because resources are needed for reproduction and growth, and this might be a primary determining factor of flowering phenology before adaptation to pollinators can take place. And this might be why most flowering was seen in the start of spring and summer in the two syndromes, with peaks occurring in spring or early spring and early summer respectively for the two different groups. Bird migrations are also an important factor that are linked to climate. And that brings me back to these exceptions that I mentioned. So in the genus Niphophia, you can see that in both specialist and generalist bird-pollinated species, most of the Niphophia flower in summer, with the exception of Niphophia ruperi. Now, when we look closer at these generalist bird-pollinated species, we see that the two that flower in summer occur at high altitudes, whereas Niphophia ruperi, which only occurs along the coast, flowers in winter or early spring. And there has been evidence and research showing that birds migrate from high altitudes to coastal areas during winter, and therefore would not make sense for Niphophia northii and coalescens to flower in winter or spring when their pollinator is not around, and would therefore make sense that they would flower in summer when their pollinators are at higher altitudes. So it is very important to also consider the impact of altitude, which may also impact the timing of flowering phenology. Then, of course, we must also consider the impacts of climate change and what that will have, the implications of that on our pollinators and the flowering species they pollinate. And if climate change disrupts the temporal partitioning of specialist species, this could have devastating consequences both for the pollinators and the plant species. So in conclusion, patterns of flowering phenology differ between generalist bird-pollinated species and specialist bird-pollinated species. Further work investigating the interactions between bird pollination class, altitude, latitude, and temporal changes in bird abundances could provide significant insights into understanding the evolution of flowering phenology and how the selective forces play out that result in flowering phenology happening at a particular time of the year. I'd like to thank my supervisor, Prof. Craig Peter, for all of his support during this project, the institutions that provided data, as well as all the citizen scientists from iNaturalist and the Virtual Museums Project who contributed their observations, the many people who provided evidence of pollinators to me, and also people who gave me access to their photographs, and then my funders, Rhodes University and the NRF, and Sandy, who funded my attendance at this conference through the Joan Range Scholarship Internship. Thank you very much for listening, and I look forward to hearing your questions.
Thank you very much, Lauren. I must say, in pollination ecology, we're really spoiled with presentations with lovely photographs, and in this case, some really nice graphs as well. So really, thank you. Thank you very much for that. Now we have, we can take 1 or 2 minutes for some quick questions. So please post them in the Q&A box. Whilst we wait for people to do that, just probably not a fair question, but you've looked in the summer rainfall parts of the country, about the winter rainfall part of the country. So the fine course, that's where I've done most of my bird pollination work. So I'm just curious what you think in terms of patterns in the Cape.
Well, there are quite a few different. It is quite a tough one because I don't know the answer, but I would think there are quite a few different factors playing out in the winter rainfall region compared to the summer rainfall region, particularly having the wet season being in the cold time of the year, which will impact growth and reproduction. And that will surely be a large player in determining when it is possible to flower or not. I expect for the specialists, it certainly will still be throughout the year because they do kind of need a nectar supply to be available to them all the time. For the journalist species, it depends on the availability of other food resources. So if there is enough food during the summer, then perhaps they would flower during the summer and maybe the pattern isn't as strong in the winter rainfall region. But I think it largely depends on the other food sources that are available.
Yeah, well done. Good answer for a tricky question. Yeah, anyway, let's not go into that discussion. We don't have time for that. I just want to see if there are any other questions. No, there are none. Then we go for a quick break and we come back at 2:30 and we've got 3 more interesting talks coming up. So please take a quick break, grab some tea or coffee or something to drink, and let's be back at 2:30. Thank you.
Thank you.
Thanks. Thank you.